We'll talk about blood typing. So this is like objective section 4 on your, if you look are looking at your objectives. So this table is, and this is out of Pearson, but if you look on Google, you can, I'm sure, find something similar. But this is a really good table, or you can make one of your own. If you write it out, it helps you remember it. So on this table, though, are some very good things that you need to know. So first of all, and since we're not in lab, unfortunately, we can't do the whole blood typing lab, which is pretty cool because a lot of people don't know their blood type. But if you go on YouTube, I promise you can find a blood typing video. And if you've ever had done your blood type, then you know what's involved. So, I mean, you're going to take a little bit of blood, and I'm going to show you some pictures of how the blood typing is done. But you're going to take a little bit of blood, and you're going to put it in a slide or in a tray that's got wells, 3 little wells in it. And one well is for anti-A serum, another well is for anti-B serum, and then the third well is for anti-D or RH serum. So this would figure out your ABO blood type and then your RH, whether you're RH positive or RH negative. So just a couple of concepts as far as blood typing is concerned. Now, if you remember way back to Biology 2113, when we talk about the plasma membrane, on the plasma membrane are certain proteins that help distinguish your cells, your body cells, from foreign body cells. And it can be from another person, or it can be a pathogen, or whatever. So on your blood cells, on your red blood cells, there's all different types of antigens. We're just talking about the ABO blood typing and the RH blood typing systems. But there's a lot of different antigens that are found on not just your blood cells, but on all of your body cells. But on your blood cells, we have 2 things that we're talking about. We're talking about the AB blood group, so whether there is the presence of A antigens, B antigens, or both A and B antigens, or if there are no antigens. Okay, so one thing you do need to know is that there's this other term. If you look here, up here at the top, antigens, you may also see called agglutinogens. So you do need to know both of those terms. Antigens is synonymous with agglutinogens. And what that means is if the presence of those antigens are on the plasma membrane, or the presence of those agglutinogens, then they can cause agglutination if certain antibodies are found in the plasma. Okay, so antigens are on the red blood cell, and that's what we're looking at. If you look at the illustration here, the antigens for type AB blood are represented by this little triangle for A and this little circle for B. So both A and B antigens are found on the plasma membrane of the cell. Now, antibodies are also referred to as agglutinins, because if there are certain antibodies in the plasma and there are certain antigens on a cell, then those agglutinins can attach to those agglutinogens and cause agglutination. It causes clumping, okay? So antibodies or agglutinins are found in the plasma, okay? Antigens or agglutinogens are found on the cell membrane. You need to remember that. So for type AB blood, both A and B antigens are found on the plasma membrane, and there are no antibodies for A and B found in the plasma. So what does this mean? This means that any blood type, A, B, AB, and even O, can donate to type AB blood, okay, recipients. All right, and then on the far right, you have the frequency in the U.S. population, and you have it even broke down by different races. So AB is the least prevalent of the blood types. My husband is type AB. It's not very common. And AB can only donate to AB, and you'll see that. So AB can receive from AB. AB can only donate to AB. Okay? So they are referred to as the universal recipient because they do not have any antibodies in the plasma. But the reason they can't donate to anybody but people with type AB blood is because both the A and the B antigen are found on the cell membrane. Okay? So type B blood. Type B blood only has the B antigen on the cell membrane. And the antibody is going to be opposite of that. Okay, so if there's type B antigen on the cell membrane, then there's going to be anti-A or A antibodies in the plasma. Okay, so if there are A antibodies in the plasma, that means that any blood type that has the A antigen cannot donate to type B blood. So type AB blood has the A antigen, so it cannot donate to type B blood. Type A blood obviously has the type A antigen, so it cannot donate to type B blood. So the only blood that type B can receive is from other type B people and type O, okay? And then the opposite is true for type A. So type A has only the A antigen, doesn't have the B antigen. So again, if the A antigen is present on the cell membrane, then the opposite antibody is going to be found in the plasma. So type A blood, people with type A blood have anti-B or B antibodies in the plasma. So anything that has got the B antigen cannot donate to people with type A blood. So people who have type A blood can only receive blood from type A and type O. Okay, and it's the second most common blood type. Then we have type O blood. So this is me. I'm type O. Type O does not have A antigens and does not have B antigens on the cell membrane. There are no antigens on the cell membrane. So if there are no antigens, then now in the plasma we have both anti-A and anti-B, or A antibodies and B antibodies. Both types of antibodies are found in the plasma. So this means that type O can only receive from other type O people. Okay? Now, type O blood, because there are no antigens on the cell membrane, then they can donate to any blood type. So they can donate to other O, they can donate to B, they can donate to A, they can donate to AB. So they are referred to as the universal donor. Okay? So that is just talking about the ABO blood typing. Now, RH is a little bit different. RH, if you have, it's just, there's two antigens, okay? So, well, there's only one antigen for RH. RH stands for rhesus. You may see D, it's the D antigen. It's the same thing. So if the RH antigen is present, then we indicate that with a positive. So I'm O positive. So this is me down at the bottom. So I have no A or B antigens on my cell membrane, but I have RH antigens. So I'm O positive. My husband, on the other hand, will go over here. My husband's over here. My husband has both A and B antigens, and then also RH antigens. So he is AB positive. Okay, and positive just stands for the RH antigen is present. Now, negative, or all that negative means, is that there is no RH antigen on the cell membrane. So for type O doesn't have A antigens, they don't have B antigens, and they don't, if they're O negative, they don't have any RH antigens. So they are O negative. Okay. AB negative, you have A antigens, you have B antigens, but you don't have the RH antigen, so you are AB negative. Okay. All right, so that's blood typing with the RH factor. And those are the two most common. So again, there are other antigens on your blood cells, but these are the two most common. And then here is something like if you were to do a blood typing in lab or something like this. This is something similar to what it would look like. So here on the left, we're just doing the anti-B, okay, or excuse me, anti-B. We're doing the ABO blood typing, okay. So here you would just prick your finger. You would put a drop of blood on the slide, a drop of blood here, and a drop of blood here. And then we have this serum. We have anti-A serum and anti-B serum. And we would just put a little drop and a little drop. And if agglutination occurs, so the way to tell agglutination is if you look at, I'm going to erase my markings up here. Okay, so if you look here with the blood on the slide up under the top one, okay, so same thing. Both of the all four of these slides, we put a drop of blood here, 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 drop of blood there, drop of blood there, drop of blood there. And then we put this serum, and this is basically just antibodies. So you're putting A antibodies in that first column and B antibodies in that second column. So if clumping occurs, and you can easily see clumping if you look at this versus this, so this second, if you're looking at the slide, the second from the top, so that circle over on the left versus the circle on the top there, the one here, there's no clumping. This one has clumping, or agglutination is what it's called. So those antibodies, those anti-A antibodies, or the A antibodies, are reacting with the A antigen on the red blood cell. Now, since this top slide is type AB, the blood is also going to react with B antibodies because there are B antigens on the cell membrane. So this person would be type AB. Now O down here, remember there are no antigens, okay? There are no antigens on the blood cells. So even though we're putting A antibodies and B antibodies, there's going to be no reaction. So there's no clumping. So we know that that person is type O. Whereas if we come over here on the right-hand side, okay, this is the same. So we have type A, this is type A. We have clumping in the A. We do not have clumping in the B, so we know that's type A. We do have clumping in this last column, which is the anti-D or RH. So we know that this person is A positive, okay, A positive. So that is the blood typing in a nutshell. Before I go real quick, on the next slide, you do have who you can give to and who can you receive from. You do need to know that. And then also note that ABO antibodies are naturally found, whereas RH are not. And I'm going to have to end because I only have 15 minutes.